extraction buffer helps to break down the cell wall and the membrane. The ethanol helps to clean the DNA and purify it. And what precipitates the DNA and proteins is the chloroform.
The first step in the protocol was to weigh 20 milligrams of dried plant tissue, in this case the ginger rhizome. Next, 600 microliters of extraction buffer were added to the microtube, and the solution was mixed with a vortex. The first centrifugation took place after waiting and preparing a twin tube to put inside the machine. The conditions were 4 degrees for 10 minutes at a speed of 11,600 RPM. Finalizing the centrifugation period, 2 phases were observed in the microtube, so the supernatant was removed and 400 microliters of laces buffer were added. Then the solution was mixed with a vortex. The sample was took to the incubation chamber, and the incubation took place at condition of 65 degrees for 20 minutes. After this, the sample was mixed by inversion, only moving up and down the solution. Continuing with the practical, the sample was transferred to ice and put it inside the laminar flow hood. Then, 500 microliters of chloroform in the isoamyl alcohol solution were piped in, and then they were mixed well by inversion. In this part, the second centrifugation took place, repeating the same temperature, time, and speed characteristics. Past the 10 minutes, the centrifugation produced 3 layers on the microtube that were very easy to see. In the top, the liquid or aqueous phase, in the middle, the debris or proteins defined, and on the bottom, the chloroform. Later on, one of the most tricky parts of the practical took place, when the team need to pipe it off the aqueous phase, taking care not to suck any part of the middle or chloroform phases. The separated aqueous phase was placed into a new labeled plastic tube. The supernatant was measured and found about 300 microliters. It was decided not to do a second wash in this time. Continuing again in the laminar flow hood, the same volume of isopropanol was added to the microtube as the recovered volume of the supernatant, consisting of 300 microliters. Also, 10 microliters of 5 molar sodium chloride were added to the microtube and mixed gently by inversion. Then, the last centrifugate of the day took place at the same 4 degrees for 5 minutes and at a different speed of 8700 RPM. Extra liquid was poured off the tube, and after checking the presence of the DNA pellet, the tube was put into the freezer.
Pipette off the liquid, being carefully not to lose the pellet with your DNA. Resuspend the pellet in 100 microliters of TE buffer plus 10 microliters of 3 mol acetate sodium plus 1 microliter of RNase. Incubate samples at 35.7 degrees for 20 minutes. ice and add 3 volumes of absolute ethanol. This equivalates to 330 microliters approximately. Then centrifugate for 3 minutes at 8700 rpm. Of the supernatant and add 100 microliters of TE buffer plus 10 microliters of 3 mol acetate sodium plus 3 volumes of absolute ethanol. Centrifugate for 3 minutes at 8700 rpm. Pipette of the supernatant and add 3 volumes of ethanol at 70%. Centrifugate for 3 minutes at 8700 rpm. Pipette of the liquid, being careful not to lose the pellet with your DNA.
The final result is a pellet of DNA of ginger.